Hello, everyone. My name is Metka Lenassi, and I would like to welcome you to this week's edition of Extracellular Vesicle Club. EV Club is an event of the International Society for Extracellular Vesicles, and it was established by Ken, but lately Clotilde and myself are also helping with hosting the event. And today, it is my great pleasure to welcome our speaker, Dirk Dittmer from the University of North Carolina School of Medicine. Dirk's background is in virology, but his interests extend also to EV biology. And today he will present his work on 3D super resolution imaging and drug loading of EVs. And I must admit, I'm really looking forward to your talk. And now over to you, Dirk.
Thank you for the invitation, of course. And I want to discuss sort of the main focus on my lab, which is the 3D super resolution imaging and some advantages and advances that we've made since the last publication. And then the second part is the applied part, where we use EVs to load with chemotherapy drugs. But what I will do is I will stop after the imaging part, and then perhaps we can have a discussion. If everyone is excited about imaging, we don't have to do the other part. Otherwise, I'll proceed. So that's the University of North Carolina right there, a lofty 30 degrees Celsius. Disclosures as required. There are folks that help us out, mostly the National Institute of Health, and then whatever conflicts are managed by the University of North Carolina. So as mentioned, I'm started out as a virologist, and so I think one of the utilities of this talk, and perhaps the only slide you have to remember is this one, the first, right? There are basically three types of vesicles that all are about 100 nanometer or 60 to 150, depending on how accurate you want to be. The first ones are liposomes, which are artificial membrane vesicles. In this case, we're going to look at Doxil, which is an approved formulation for the treatment of Kaposi sarcoma, whereby doxorubicin, a drug that causes heart disease, that as an off-target has heart toxicity, is loaded into liposomes. And these are characterized by a single lipid, or at most 3 lipids in the membrane. Then there are the EVs that obviously I don't have to introduce. And then there are the viruses, which are like EVs, except they typically have an inner capsid core that protects either DNA or RNA. And their membrane composition is typically simpler and enriched for just those viral proteins that make up the receptor. So if you look at SARS coronavirus 2, there are only 2 proteins on the vesicle surface, but the virus itself has a capsid, it has RNA, it has a lipid membrane. And so that's how we thought that EVs are just like the poor person's version of a virus, which will get me cut off really quick here. This is a review that Nancy Raptrop and I wrote a while ago, in that because viruses exocytose from the cell, they use many of the same pathways as EVs. And in fact, they have used EVs to utilize them for their own purpose, right? The most important part is about here, that's hepatitis A virus, which if you see it in the fecal secretion, only has the capsid and no more lipid membrane. But if you see it circulating in the body, is encapsulated in EVs. And that's the mechanism how this virus shields itself from the antibody response. That's work mostly of Stan Lemon here at UNC Chapel Hill. And there's Epstein-Barr virus, which incorporates a whole bunch of things. There's KSHV, which is what we will work on. And there's HIV, which sort of has 3 mechanisms to get into uninfected cells. One is the virus itself matures, and it has everything. And then you can see this maturation step, which is the target of the protease inhibitors. There's HIV TAT, which is a secreted protein that has a 20 amino acid sequence that allows it as a protein to translocate membrane. And there is HIV-NEF, which we and others have shown is encapsulated in EVs and thereby moves to uninfected cells, and viruses like EVs because it gets them into cells that don't have the viral receptor. So first question on every EV talk is, what are we really working with? That's our purification platform. We start with purified media, we do filtering steps, and then we use tangential flow filtration, which we have published, which gets us a highly enriched fraction, about 1/10 the volume. And of course, it gets rid of anything that's less than 750 kilodalton, which are most of the soluble factors. We precipitate the virus with PEG, rotate, incubate. Here, we're going to get rid of any RNAs and DNAs that were co-purified because we're treating them with RNAs and DNAs. And we use a captocore column to, again, retain anything and everything that is less than 800 kilodalton. And that allows us to generate EVs that have never been subjected to G-forces more than like 1,000, right? So it's very different than ultracentrifugation, but essential because we want to keep the EVs as native as possible. We have expanded that to a tiny little bioreactor that we're playing with. It's a lot of work for not that much volume, but it allows us to get about 10 to the 13 labeled EVs per week at a concentration of 10 to the 11 per mil. So every week, that's what we start with, typically on a Tuesday. And this is from the recently published paper. It shows that our EVs are labeled with either CD81 M-cherry, CD63, GFP, or both. They look like EVs. The M-cherry ones, you can just, they're red. You don't have to do anything special. They look like EVs under EM, and they have all the markers for which our Western blots work. They're fusion competent. That's sort of the standard assay. You put in a dye and you show that the dye is transferred. And we expanded that in work that's not published to show that we can take these EVs, and here we're adding a CD81 affinity purification step, and actually transfuse them into monkeys. And we can see that they're taken up by PBMCs. We can follow the GFP CD81 or the labeled EVs, and they're taken up by endothelial cells on the website. So anything and everything that we do in our purification procedure preserve the biological activity. We can show, and this is earlier work, almost 10 years ago now, okay, that we can separate the EVs from the viruses because, as many of you know, most EVs can be blocked by annexin 5, which doesn't block the viruses. And the EVs are not blocked by a concentration of heparin that would block the viruses. Now, I might add as sort of a caveat, each virus has its own preferred heparin concentration. So if you go high enough, you can block it. But this virus that we work on is blocked at the concentration of heparin where the EVs are not. Now, since we have so many EVs and they're so pure, we were able to do cryo-EM. And these are some of our cryo-EM structures. They're similar to what other peoples have shown. The nice thing, it shows EVs are really round. But what I want to stress, and this is the first glimpse of what I'm going to talk about, is that there are two types of membranes. There are the smooth ones, that's the white arrows, and then there are membranes that are full of protrusions, which we know are proteins. Now, these are unordered, so we can't crystallize them and get 3D structures. But what we can see very clearly is that there are regions that are less order, that have less proteins, and regions that have more proteins. And that's really what we wanted to go after in super resolution microscopy, in particular D-storm. This is an introduction to D-storm, which basically says that the diffraction limit of light is about 200 nanometer. The resolution will then be 100 nanometers, and our EVs are much less. Rather, the limit of resolution is 200 nanometers, and the EVs are less. So you can't separate any two EVs by conventional microscopy, but you can do it by super resolution microscopy in this kind. This is D-storm, where basically rather than having the fluorophore on all the time, it emits short bursts, and these can be mathematically processed to give this pattern that generates a level of resolution that other techniques simply don't have. This is how this looks like. Again, we can see an EV. This is labeled simply with a membrane dye, which is very bright. And we can do that in 2 dimensions, and we can also do that in 3 dimensions. What we measure by EV matches what we get by NTA. And so that's an advantage. It's always a little bigger than transmission EM, because transmission EM dehydrates the EV. So these EVs are in PBS. There's nothing special that we do to them. They're just labeled with a particular dye. And I hope you can see that, but you can actually resolve them in 3D. And resolving them in 3D allows us to then start to localize proteins and regions. They're colored just by randomly, but that's kind of the signal you get. You still get a signal that requires a fair bit of computation, but it allows you to get insight under physiological conditions that you won't get otherwise. This is the dye, and now we're starting to do a statistical analysis. And so I simulated, we're all on the unit sphere, 2 types of EVs. One where the dye signal is truly random, and you can see that here. And one where the dye signal is intentionally localized on the pole. You can see that here, and you can see that here. What we measure looks like this, and these are just 3D representation, in that there is a localization, but then we also very nicely see this donut shape that many of you seen, and that is really characteristic of the super resolution, because we can distinguish between the dye and the membrane, which is about 40 nanometers or 20 nanometers, depending on which textbook or how you count that, and then the inner part of the EV, which clearly is not filled with lipids and is not filled with dye. And all our EVs are donuts. Here are 6 of them. They look different depending on the staining, right? So this is not the resolution of cryo-EM, but it allows us to obtain lots and lots of images that have similar properties. And when you do that, you can start zooming through the Z part. So this might be a little bit too much mathematics, but you can see that you have an EV which is round. It's actually not completely round because there is some distortion that we adjust for. And then you can make slices, these Z stacks is what they're calling them in microscopy, that have a diameter in nanometers, and we're going to slice through, and at the same time this can be used as a projection, except in the projection you average them all, but as we slice you can see this kind of images. So here we're slicing, this is exactly the equator of the EV, and then this is the equator minus 90 nanometers and plus 90 nanometers, and you can see the donut hole, and you can also see that the dye is unevenly distributed. There seems to be a lot here and very little here. And you can see that this patch that starts at -19 goes through the equator and ends up at +19. That is, it's about 40 nanometer in patch, which now it gets into differential geometry, but that's the easiest to think is there's a ball and then on top of it is a 40 nanometer enrichment. We can use that information to then run whatever our favorite statistical tools are, and that's actually one of the research directions that we're taking in the lab. We can triangulate things, we can do in fact correlation, which shows that there is a big cluster here and a big cluster here, which you don't see in a random sphere. In the random sphere, you have small clusters because it's random, but they're on the diagonal. Whereas in the polar sphere, where we now intentionally build clusters, you can see the same kind of patterns, and you can just look at the size of these squares that we observe. And that's one of the arguments to say that there are membrane domains on individual EVs that we're observing. That's how they look. Now we're going into two colors. There is aprochromatic correction that we do to get the red and the green overlap, but that's nowhere near as when we're now trying to localize proteins. So before I told you membranes form membrane domains on individual EVs as probed by a lipid dye. Now I'm going to tell you that we can localize proteins, and in this particular case, tetraspanins, on the surface of a single EV. And that's shown here. This is just two dyes, red and green. And you can see they overlap except for the achromatic thing that we need to correct for. Here we have CM green and M cherry, which always sits on one side and never all around. And here what you can see is the CM red and CD81 are further apart, right? You can see here they're touching in the middle panel. They're quite apart on the right panel. And that's explained here, the red and green overlap. We have our single enrichment of CD81 and cherry here, but if we use an antibody, which itself is about 100 nanometers, we're pushing the Alexa 488 floor away from the EV, and that's when you see this very nicely marked distinction. So both by antibody or by direct labeling, we can show that the tetraspanins sit on one direction of the EV. And one of the things that we're really actively pursuing right now is to localize more than one tetraspanin. Here we have CD81 and CD9. And this one is particularly pretty because they're right on top of each other. But even here, you can see most of the EVs, the two tetraspanins are right next to each other, separated from the dye. And so that brings up this fluid mosaic model. That's the second slide that you have to remember, which is how we think that EVs have tetraspanin microdomains. Fluid mosaic model is a term that I learned when I was back in college, which showed that basically no membrane ever is random. The cell membrane certainly is not random. They're membrane domains, and you can disturb the fluidity of the membrane by the amount of cholesterol. What we have shown now is that the same applies to EVs. So no matter where they come from, and we don't know that right now, they nevertheless have membrane that have patches, and at least the tetraspanins seem to like to hang together, which makes sense because tetraspanin by definition or by nature are multimeres. Now it looks like that even heterogenic interactions between the tetraspanins are the norm on EVs. I'm going to stop here so we can discuss. And then if there is more time, we can go to the second part, but I want to make sure that I'm answering any and all questions. So if you want to unmute yourself.
Do you know that I think that you can just finish with everything that you have to say? People are
Okay.
still typing the questions, and yeah, and the questions will be typed, so everyone will remember.
Okay, so David, just hang on there and type
Okay,
in.
there is one. I mean,
Okay,
we could
David.
okay, S to unmute. Okay.
I'm sorry.
For a quick question,
thank you.
go ahead.
Thank you. Thanks. Very, very interesting. Maybe I missed it, but so actually one of the questions I had was on how exactly the EVs were immobilized, because I think this might affect their circularity if they're immobilized on a surface. And then maybe the second question is I envision the EVs as being thermally agitated and wiggling around, and I guess there is some averaging through your storm imaging, but how much? So how does that overlap with the imaging, and whether that actually diffuses your imaging, your precision, your accuracy in your imaging?
Right. So there are two things. One is the images I showed you, we simply dropped them on regular charge slides. So there is no affinity capture in this work. And we can show by D-Storm that the EVs that we image are actually sort of in the middle of the workable distance, right? This is not TRIF or turf where you go in from the bottom and you measure within 5 or 10 to 20 nanometers off the slide. This is D-Storm where we zoom through probably 200 to 400 nanometer space to find one that's isolated.
Okay, so
you with me there?
So they're on a charged slide, but so they're not flattening out on the slide, or is there any? Is there any, or how do you? But how, and why don't they diffuse away then? Because they are
right. So that has to do with the time frame at which we obtain the images. So if we have a really bad fluorophore like GFP and RFP, we have to record the data over a longer period of time. With D-Storm fluorophores and the crazy big lasers that the ONI people put into their microscope, we don't see that. We can record in a small amount of time a lot of images if we can label the EVs bright. And that determines, as you said, the degree of resolution. Now, I told my lab I want to put the microscope in a cold room, but they refused to go there. We have it, we'll put it on a cold thing to control for temperature, and we have it on a vibration table. And the buffer that we're in has a fair amount of glycerol to make the density, right, that dampens. So there are lots of these little things, but none of it damages the EVs so much that we couldn't inject them in mice afterwards and get a phenotype.
And so just so how long is D-Storm imaging, or how long does it take to get one of these images?
Let's see, Alara, are you on? Hold on, I get you the exact number as of last week.
So maybe we can put that in the chat.
Yeah, I will answer. Yeah, Laura will put the answer in the chat. It's not that perfect. Much. It's not hours.
So thank you, David. There were two questions that I will just read out because I think that otherwise it will get messy. So do you see multiple, so from James, do you see multiple tetraspanins on your EVs, or are there particular pairings or groupings?
We have a few EVs where we can see multiple tetraspanins. That is, what we see is the tetraspanin signal present at opposite poles, like here and here, and the EV is in the middle, of CD81 labeled and CD81 GFP. These are relatively rare, and I don't know why. Also, I think when I talk about tetraspanins, remember, these are not individual molecules. There are some X-ray and cryostructures of tetraspanins, and there are someone correct me, but they're at least tetramers, if not more. So they're still really big patches. We're nowhere, this is not single resolution, right? We are requiring molecules to be tetramers or octamers to be sorted into one D-storm signal.
Okay, perfect. And then another question by Li Fang. So can you compare the storm versus ONI to image EVs?
Yeah, we are using an ONI machine. I just don't want to use the brand name in a talk.
Okay, that was easy. Do you see any application of, so this was from Same. Do you see any application of this storm imaging for EV cargo visualization going beyond surface markers?
Yes, that's the grant I'm writing.
Okay.
Yes, you can do that. I think you can certainly, I will show you pictures, you can show using a nucleic acid dye, which is doxorubicin, that the EVs are loaded with nucleic acid. But again, we're not at single molecule resolution. If you have a cargo that is an octamer that labels really well with the super bright GFP that's on the inside, you might be able to localize this. If you have a single molecule, you have to go not in just 100 to 120 nanometer EVs, you have to go to 400 nanometer vesicles to make the hole in the donut really big. And then you can try those. And obviously that's one of the areas that we're pursuing.
Okay. So I think that now we can continue with the other part of the talk, and there'll be plenty of time also in the end for any other questions on both topics. So
okay, now let me, no, I think that's important.
You introduced a new system to the EV Club, so that's great.
Well, I think it's important that, right, you guys are the experts in the field, and we need to have time and opportunity to go into experimental details, which you don't often get when you give just an overview talk. So this one, we were trying to use EV as a scaffold for chemotherapeutic agents. And those didn't come out by random, but they come because we work on a virus called Kaposi sarcoma. It's caused by a virus. It's a tumor of endothelial cells. It's the most angiogenic tumor in humans. And it's treated with liposomal doxorubicin, that is an artificial liposome formulation of the drug. And what you can see here is simply the brown parts. Here is the tumor, and these are the blood vessels. So it's very clear that anything you put into a blood vessel will end up in the tumor, and the tumor is leaky, which is why you then get this extravagation of red blood cells. We had also shown that in this particular case, and that's the trick we're using for all of our microRNA work, there are only 5 microRNA that the virus makes that account for 75 of all molecules in the EVs. And the 2 other ones are the lineage marker for endothelial cells, that's miR-121 and miR-122, and a bunch of smaller ones. But the virus itself makes the overwhelming amount of microRNAs. And we have shown that using, again, affinity purified EVs. I'm going to walk you through this. This raw data. I really like to show raw data. Nothing in uninfected cells, lots of in virally infected cells. This one is EV from a patient. This is CD63 purified EVs from an uninfected cells and infected cell culture and infected patients. And these are the actually 12 microRNAs. And then GAPDH, of course, is not as an mRNA much in the EVs, and neither is this protein. And we use digital PCR to do this. And the advantage is that digital PCR, of course, counts individual molecules, right? That's the theme of my lab. We want to look at individual events. And you can see lots of microRNAs in the cell. but you can clearly see microRNAs in the EV fractionation that's affinity purified. And because we are counting individual dots here and we're counting individual EVs before, we can estimate about 10 plus minus 10 molecules of MIR-K12, 10 per EV. So if anyone ever asks you how many microRNAs are in an EV, it's about 10 of any one. And since we have 5 of them, we are anywhere between 50 to 100 microRNA molecules per EV. And what we've been able to show is use this didoxorubicin, which is a nucleic acid intercalating dye, which is also extremely hydrophobic and will partition into membrane to load EVs, right? People have done this with artificial liposomes. So we were sure when we started, we would at least get the dye into the membrane. And the dye is cool because it's red fluorescent. It's also rather toxic, which is why it's a cancer therapy. So you don't want to drink it. But what we then observed was that we had much more drug within the same number of EVs, and they're the same size, than with the currently best clinical formulation. And this one was calculated by mass spec under clear. And moreover, what we observed, you can just take the EVs, and then every day you run them through our captocore column to separate out the dye that's not in the EVs, and you keep the EVs. And what you noticed is what has been observed in the literature. If you take the pharmaceutical composition, the dye rapidly moves in and out of the liposome. And so if you remove the dye every day, then it depletes rapidly, whereas in the EV, it stays. So it seems to be retained by something other than the lipophilic character. And you can see that it's temperature dependent. So if we reduce the temperature, everything slows down. These EVs do what they're supposed to do, in that if you use normal EVs and put them on cells, nothing happens. If we use our EVs, we induce DNA damage, which we measure by gamma H2X phosphorylation. Doxil does the same, and free drug does the same. So the drug in our EVs is the same as you can buy in the shelf. We think the EVs kill a little bit faster. That's on the right, this blue line. It is significant, but whether it's clinical significant, I don't know. We did this, in fact, and here I have to tell you something about Kaposi sarcoma. There is an oral form, which you can see in the inside of your mouth. And the way that's treated in the clinic is you inject the doxil drug directly in the lesion. I actually tried that on my cat when she was dying, but it didn't work. But that's how it works in the clinic. And so we do the same thing. We take the doxil and the EV and put them in a plug on the back of the mouse. And here we can really see the change in phenotype. You have, this is the plug. If you do PBS, you have mouse cells migrating into the plug. Wedge F makes lots of CD34 positive endothelial cells. That's neoangiogenesis. The viral ones themselves are extremely angiogenic. So part of the transforming phenotype of KSHV is mediated by EV. But if we take these same very angiogenic ones and we fill them with dox, we can reduce the amount of angiogenesis. And so we don't know what the targeting is of the EV. We just say, hey, we take whatever the virus makes as a bad thing, we put the drug in, and it will kill whatever, wherever they go. And we also can put in 2 different drugs. Paclitaxel is second line for Kaposi sarcoma. We can administer them together. What that does to systemic toxicity, I don't know. And then we wanted to see, and I'm almost done here, what is it in the EV that retains the doxorubicin much, much better than just the lipophilic membrane, right, which is the doxil formulation? And we thought, well, maybe it is because unlike liposomes, which only have lipids, the EVs have lipids, and then insides are nucleic acids and proteins. Now, we couldn't get rid of the proteins because cells need them. But we could get rid of most of the nucleic acids because we can make microRNA negative EVs. Turns out that dicer is required to make microRNAs, but it is dispensable in tissue culture. So we're taking isogenic cell lines. These are now run-of-the-mill HEC293 cells. We knock out dicer using a Talon construct that is published. We make EVs that have all the same markers. So you can make EVs that don't have microRNAs. And then if we look in the dicer knockout RNA, EVs, there simply aren't any microRNAs. And this is again a qPCR array. There's a spike in control, and then these are the viral ones, and this is the rest of the human ones. And so then we can compare microRNA positive and microRNA negative EVs. And that's shown here by flow cytometry, because I told you that the doxorubicin is in fact fluorescence in the PE channel. If we have unloaded EVs, they don't fluoresce. If we have wild type EVs, they fluoresce a lot. If we use the dicer knockout, we reduce mean fluorescence by about a log. This is log scale here. And then again, we can do that quantitation over here. And so the conclusion slide is quite nice, I think. I think we can use doxorubicin, which people have used in an artificial liposome, to load EVs. Now, at least 80 to 90% of the doxorubicin will actually bind to the microRNAs, because if we remove the microRNAs, we lose the signal. Now, there's still 20% left. I don't know if that's in the membrane. I don't know if it's bound to protein, or I don't know if it's bound to tRNAs. You can't really make a tRNA-free cell easily. But what I do know is if doxorubicin is given the choice, it will distribute to each membrane, but the moment it hits the microRNAs, it doesn't come out of the exosome anymore. And so you generate very stable, natural exosomes that retain twice the drug loading than anything that's on the market. And with that, I just want to acknowledge my lab. A lot of people work on EVs. A lot of people work on the virus. Even more people worked on SARS-CoV-2 in 2020. But now we're all vaccinated, and so we're having quite a bit of fun. And with that, I'm happy to take questions. I'm going to leave the screen share on so we can flip back and forth.
Yeah, this is great. Thank you for also this other part of your talk. I think that the whole story was very inspiring and interesting. And so for everyone, please just type your questions in the chat box. And there is a question already waiting for you, which Shalini asked me to ask because he doesn't have a speaker. So how do you prepare your EV sample for this storm? How do you remove unbound dyes or antibody, and how you make sure that the signals are coming from the EVs, not the unbound dye or antibodies? So,
okay, so one is you can incorporate. Is very simple. We use Captocore 800. You mix the EVs with dye, and then you run them through the Captocore column, at which point the free dye stays in the column. And so that dye then tells us these are the EVs. And then for the protein signals, we discard any protein signal that is not localized to the lipophilic EV. So if there were aggregates of proteins or antibodies, they would be green, but they wouldn't be near any red. And the software of the company that shall be not named does that very nicely. And then once we have a particle where they're all within 500 nanometers, or yes, then we go in and we refine the image to the 3D, and then we can localize the proteins on one side of the EV.
So did you get rid of DNA on the surface of EVs?
Yes, our purification pipeline uses a DNA step and an RNA step.
Mm-hmm.
Yeah.
How do you so with DNA this, or why did you ever try to also look at DNA corona or what is bound on the surface of EVs?
Yes, this will get me expelled because I don't believe in the DNA corona.
Okay.
And that's simply because of the discussion we have in the virology field. And that very similarly, there were lots of observations of microRNAs attached to viruses in viruses. And over time, people were able to purify them away. Now, I have to qualify that because it's quite possible that you have EVs that attract DNA, because we now know that you have circulating free DNA, right? ctRNA, which is used in cancer diagnosis. So yes, there are probably electrostatic interactions between free DNA and free EVs. But I think these are random. So if we get rid of them, that would be better.
Mm-hmm. Well, there can, I'm a bit of a believer in. We're just trying to get a paper out on that. So, no.
Okay, well then just say he's a virologist. He doesn't know better.
No, I'm joking, but of course it can be non-specific binding. It's not supposed. It doesn't have to be a specific binding, but there are. There, at least in our experiments, but you can then comment on the paper and we can discuss later.
That's actually going back to David's question, right? Maybe the reason that our EVs are not flittering around is because they're decorated with DNA, which of course would introduce tremendous drag, right? Yes.
Okay, perfect. So Sasha, do you want to unmute yourself? Sasha Koeskin.
Hi. Yeah, my question is just I think Dox is positively charged. Do you think it's the physchem properties of this particular active are showing this behavior, whereas other pharmaceutical actives would not?
Other glass plates, or which behavior now?
The microRNA stabilization.
microRNAs of doxorubicin, yes. I think doxorubicin has been shown to bind to nucleic acids. We're obviously now trying every drug we can get our hands on to get a better understanding of that. I mean, the reason we picked it is because it's a known DNA, like one of the best DNA intercalators that has made it to the clinic.
So you probably have not seen this behavior with other actives you looked at?
Not as a lot. I think we didn't really spend a lot of time trying to find a drug that doesn't bind, because there are lots of drugs that don't bind DNA, but they still have to partition through the membrane. So honestly, that's a PhD thesis waiting to happen. Because you can imagine that you can make derivatives that more or less bind to microRNAs, which is something pretty cool. Again, okay. Yes, I do look for postdocs.
Okay, thank you. And David, you have another question? Just go ahead.
Yeah, well, thank you for this fascinating talk. Actually, I have two more questions. Maybe I'll slip another one in here. And so the first one, in the beginning of your talk, you made some comments about that you didn't use high G centrifugation because this would preserve EVs. So I mean, I don't use high. We use a bit of centrifugation. It's not our expertise, but having heard of this being the gold standard in the EV field historically, it'd be interesting to hear more about that. And then my second question, if I may, is then about your last slide where you showed that with dicer you could, by adding dicer, you reduce the loading of the doxorubicin within EVs. So have you tried the opposite by trying to add RNAs into liposomes to try to retain doxorubicin, which could have quite important clinical significance if that were the case?
Yeah, I think there is an awful lot of pharmaceutical or school of pharmacy research trying to augment liposomes with nanoparticles. I think they're way better than me trying to achieve that. I just want to understand how it goes into exosomes. And I do think exosomes, by virtue of having proteins, and many people believe these proteins are tumor specific and have targeting properties. I don't have to know what the targeting properties of the EVs is. I just need to show that they go where the metastasis will happen, and then I poison them right out of the patient. So we're a little lazy there.
Okay, and about the purification process and the centrifugation?
Yeah, so there are two things. One is when people do that 100,000 K spin down, I think that's really hard on the EVs. And that's why we don't want to do that. I think when people do flotation gradients, that's a different story. And you can use cushions, and we've done those experiments. They're just very cumbersome. Remember, we start with a liter every Tuesday, and it has to be done by Thursday, right? And so the way you can scale this tangential flow filtration, we could start the bioreactor I showed scales up to 20 liters, and we would still only need a week. So that's why our processes take out the centrifugation. And the other part, of course, is we can use this process in a biosafety level 3 lab when we work with viruses, including HIV. So whether it's superior necessarily to gradient and other centrifugation methods in terms of biochemical purity, I don't know. But I think the function is better preserved. And then obviously, if you're really desperate, you can take what comes out of the captor core and run it either over another column, or you can run it over a flotation. I would not spin them down. We have some success with centricon filters if we really need to reduce volume, but those also don't get spun at 100,000 G.
But is there experimental data showing that, or have you seen that there's degradation of EVs? Because I haven't seen all this documentation of that. Or
have you seen that? Yes, we actually published a paper on that looked by EM, and there are much more broken pieces, and also there was much more aggregates that have the same density as EVs but are protein aggregates that come down not with the flotation but with the 100,000 K step. Now, and so I think the key part here is that we're not just dumping PEG into tissue culture media. We have the TFF step that removes any protein up to 750 kilodalton. So those are the ones that form aggregates. And it's only afterwards that we add the PEG. I think if you do TFF and then the 100,000 K spin, you would probably get exactly the same preparation. But if you just do the spin out of supernatant, you get aggregates.
Okay, thanks so much.
Yeah, thank you. Just a quick question: how do you remove PEG, or is PEG still then bound to the EVs when you look at them?
Yeah, that's where the capture core 700 comes, which is a size and charge resin that was actually developed to make influenza vaccine. Like most of the things we just steal from what virologists have been doing, and that gets rid of the PEG, and it gets rid very nicely of BSA, for instance.
Okay, nice. Guoping, are you here?
Yeah.
Okay, perfect.
So,
go ahead.
A quick question about, could you try to use your DStorm system platform to image the location of doxorubicin after loading? Like, because I think doxorubicin can be conjugated to a lot of other chemicals, or it can be modified using azide groups, something like that.
I think that's an awesome idea, and we have not. One thing with DStorm, which is what most people don't tell you, is that you need to have that DStorm pulsing activity, which most regular fluorophores don't have. And so we would have to conjugate, just as you said. It's a really cool experiment. We haven't pursued that.
I'm interested in the doxorubicin loading, and effectively, if there is a microRNA in the particle that retain the doxorubicin, how does this become an effective drug, and how do you deliver that to the target cells? It's not a free doxorubicin anymore. It's complex already to nucleotides.
Right. Well, all we know, what I'm showing, is the EVs fuse with the target cells. And then I don't know what happens, but 40 minutes later, the target cells die of DNA damage, which is the mechanism of doxorubicin. So we don't know the mechanism, we just know it works.
Oh, I might have missed this part in the talk, but did you guys load exogenous doxorubicin into the EVs?
Yes.
What was the loading strategy, if so?
We mix it in. We can actually show that in one experiment, we mix doxo, which is liposomal doxorubicin, with EVs, and it will partition, right? The thing about doxorubicin, it really readily partitions in and out of membrane. If you think about the EV as being membrane and aqueous in the middle, half the time the doxorubicin will stick to the membrane and go right back out in the solution, and the other half of the time it will go into the other direction. And so we haven't optimized any of this, and certainly you can inject, you can charge, you can transfect, you can do all sorts of things to get more doxorubicin. But because it binds the microRNAs, it introduces an element of irreversibility. So if you just incubate them long time, eventually you saturate, and that saturation is determined by the amount of microRNAs in the EV. It's not fast, it's nowhere near production ready, but it shows the proof of principle.
I will just read out the question. You show that EVs from dicer knockout cells have decreased amounts of doxorubicin. Did you somehow validate this is due to doxorubicin binding to microRNAs? So
well, we show that the EVs that are released from the doxa knockout cells, which we purify same way we purify all EVs, no longer have microRNAs by qPCR array. That's one of the slides. I might have gone way too fast through that.
okay. And Sasha Kreskin has a short question. Okay, I'll just read it out. Again, about the PEG. Obviously, the PEG, everybody noticed the PEG is present, and she just wanted to know, how do you check that PEG is gone?
How do I check that the PEG is gone?
Well,
so it depends on the application, right? So one way to show it is that we inject the EVs into either mice or monkeys. They really don't care. So there is no acute biological reaction to the PEG. The other part is, depending on how pure we want the EVs, we might add a CD81 affinity purification step afterwards. And again, the PEG gets diluted and it gets removed through the captor core column, which is shown to retain the PEG. But if we want really, really pure ones, we will add an affinity purification step and just get those EVs that express high levels of CD81.
Hi, Dirk. Nice to see you.
Good to see you.
Beautiful paper. I read it. The super resolution EVs. You mentioned in your talk is about 50 to 100 microRNAs in EVs. How do you calculate that?
Okay, that is the famous back of the envelope calculation. There. So this is in Plus Pathogen 2019. But basically we load EVs into these digital PCR chips, which have which count exactly the amount of molecules that are in 10 microliter. And then we count exactly the amount of EVs that are in 10 microliters. And that's the ratio.
I see. Okay.
And this is just one microRNA. Then I know that probably they're at most 12, and the other ones are minuscule, so they're multiplied by 12.
Okay, okay. Thanks.
So it's 100 plus minus 200 for variation, probably. But there are a lot of people that say there's no more than 1 microRNA per EV, and this experiment disproves them, which is why we did it.
No, this is a great experiment. In our studies, we do find multiple, like you are saying. There is only one concern that I have, that is your second part of the talk, that we find only about 10 to 20% of the total RNA is microRNA. Others are tRNA, and
Yes, and we find exactly the same in that there are lots of tRNAs.
And so that means,
and so that's why the signal here, if you look at this red peak, it doesn't go back to 0, right? It stays there. And I think this difference is the microRNA, and this difference is everything else.
Yes.
But we were just happy to show that the drug binds to 1 nucleic acid species. And that's the big difference, because none of the commercial liposomes have anything in there, right? They just have water in there and lipids. I think the EVs are better, or if someone mentioned, if you would transfect microRNAs into liposomes, you could disturb the equilibrium that way. But we're not pharmacologists.
Thank you. Great talk.
Okay. So I think that we're done with the questions. And Dirk, I would just like to thank you again for taking your time for this, to present this excellent work. And it was a great discussion. And I would also like to thank all the attendees that joined us today. And of course, you're also welcome to join us next week when we will discuss another topic important for EVs. So thank you all, and have a great afternoon, morning, or whichever time of the day it is where you are.
All right. Thank you, everyone, for listening.
Yes, thank you. Bye-bye.